Okay, so welcome back to this next video in which we are discussing multi-drug transporters. Okay, right. So in this video what we're going to turn our attention to is beginning our study of each of these 5 separate families of multi-drug transporters in turn. Okay, and we're going to begin with the family of ABC transporters. Okay, now before we begin, what I should say is that the family of ABC transporters is a massive, absolutely massive family of primary active transporters generally. Okay, now not all ABC transporters are actually going to be multi-drug transporters. There are a subset of ABC transporters that are actually multi-drug transporters. What I'm going to start off with is a discussion of ABC transporters in general. Okay, so what I'm going to start off with is a discussion of the structure of these things, firstly. Okay, and that applies for many ABC transporters, not just the multi-drug transporter ABC transporters. Okay, we're then also going to look at the mechanism by which they work. Now, when we describe the mechanism, we're specifically going to look at an exporter. Okay, so we're going to look at a primary active transporter which is exporting something out of a cell. Okay, because all of the multi-drug transporters are going to be exporters. Okay, however, certain ABC transporters will be importers as well. The final thing to say is that this is not just a group of proteins that is found solely in humans. This is found in loads and loads of different kingdoms of life. You find them in bacteria, plants, humans. Okay, so it's a vast area of proteins basically that's conserved throughout the entire kingdoms of life. Okay, right. So the first thing to say is that ABC transporters, the proteins which make ABC transporters, fall into 2 main categories. Okay, so let me outline these 2 main categories. The 2 main categories is some ABC transporter proteins are what are known as full transporters. Okay, and that means that this protein on its own is capable of being an active transporter. And then other ABC proteins are what are known as half transporters. And this means that these proteins on their own are not capable of being an actual active transporter. Instead, what has to happen is they have to dimerize together with another identical ABC protein in order to actually form a functional active transporter. Okay, so let me start off then with the structure of a full transporter, and then I'll show you the structure of a half transporter, and then we'll have a look at the mechanism by which these things work. Okay, so full transporter then. So I'm going to draw 2 parallel lines here, and these 2 parallel lines are going to now represent the inner leaflet of the phospholipid bilayer and the outer leaflet of the phospholipid bilayer. Okay, so this is the extracellular fluid compartment here, and this is the cytoplasm compartment down here. Okay, so this is the cell membrane. I'm just drawing the 2 separate layers of phospholipids that make up the cell membrane. Okay, right. I'm now going to draw the polypeptide then for one of these full transporter ABC proteins. Okay, so it's a polypeptide, so it will have an amino terminus. So here's its amino terminus. Then this line is going to represent the polymer of amino acids. So this represents amino acid after amino acid after amino acid. So it has the first membrane spanning alpha helix, a second membrane spanning alpha helix, a third, a fourth, a fifth, a sixth. Okay, and that's the first cluster of 6 membrane spanning alpha helices. Okay, then you have this special domain which I'm going to draw here. Okay, which is known as the nucleotide binding domain 1, which I'll label up in a moment. Then you have another cluster of 6 membrane spanning alpha helices. Okay, like so. And then you have another one of these nucleotide binding domains here, which is known as nucleotide binding domain number 2, and then the C-terminus, which is also on the cytoplasmic side. Okay, so let's highlight up a few of the important domains here. So here then, in this first cluster of 6 membrane spanning alpha helices, I've coloured these all in now blue. Now, this cluster of 6 membrane spanning alpha helices, this is known as the transmembrane domain 1, which is abbreviated as TMD1. So this stands for transmembrane. Okay, that's the TM, so the T is for trans, the M is for membrane, and then the domain is the D. Okay, so TMD stands for transmembrane domain. Okay, Then you've got another one of these clusters of 6 membrane spanning alpha helices here, and that's known as the transmembrane domain TMD2. Okay, right. Now, it's these two transmembrane domains together that are actually going to form the route by which the molecule, the substrate molecule, can move from the cytoplasmic aspect to the extracellular aspect. So the substrate is going to move by interacting with these two transmembrane domains. So these are actually going to end up clustered together, and they're going to form the portion that will actually move the substrate molecule across, as we'll see later on. Okay, right. Then the other important domains on this picture are these little structures here, which I'm going to colour in orange. So there's one, and here is the second one here. Okay, and you'll notice that I have put two little special binding sites on each of them. And these are going to be binding sites later on for ATP, okay? And these are what are known as the nucleotide binding domains. So this is the nucleotide binding domain 1, and this is the nucleotide binding domain 2, the NBD2, okay? So NBD stands for nucleotide, that's the N, and then it's the nucleotide binding domain, okay? Now, the nucleotide binding domains have another name, okay, an older name. The older name for the nucleotide binding domains, which very rarely does anyone use anymore, but it's important for understanding the origin of the name for the ABC transporters. The old name for the nucleotide binding domain is to call it the ATP binding cassette, okay? And that's the origin of these transporters being known as the ATP binding cassette transporters, because they have these ATP binding cassettes. So very occasionally, instead of seeing these special domains known as NBD1 and NBD2, you can see them called ABC1 and ABC2 respectively. Okay, so that's the structure of a full ABC transporter. So loads of examples of ABC transporters have a structure like this, and they are functional as a primary active transporter on their own. Okay, however, many ABC transporters do not have this structure, and instead have a structure known as a half transporter. Okay, so let me show you the structure of a half transporter. So many other ABC transporters only have effectively half of this structure. Okay, so they have one transmembrane domain, one cluster of 6 membrane spanning alpha helices, here's their N-terminus, and then after that cluster of 6 membrane spanning alpha helices, they then have one nucleotide binding domain before they then end. Okay, so this then is their only transmembrane domain, and this is their only nucleotide binding domain here. Okay, so we'll colour code it in again. So here is the cluster of 6 membrane spanning alpha helices in blue, and here in orange is our nucleotide binding domain with those two binding sites for ATP. Okay, right, so within the massive great family of ABC transporters, some of them look like this, some of them look like this. This is functional on its own as a primary active transporter, this is not. This has to dimerize into what's known as a homodimer. Okay, so it has to get together with another protein that is the same as it. That's what homo means, it means the same. Okay, so it has to get together with another protein that's the same as it, and then that complex, that dimeric complex, is then a functional primary active transporter. Okay, right. So, what we're now going to turn our attention to is how these things work, or at least what is understood of how these things work. Okay, now, for the basis of drawing this, I'm going to assume that we are working with an exporter, okay, because all of our multi-drug transporter, ABC transporters, are going to be exporters. And I'm also going to assume that we're working with a half transporter, and the reason I'm doing that is simply because it will make drawing the picture easier. Because if I draw half transporters, I can get away with having it in two halves. Whereas if I have to draw this, you will expect me to connect everything up in this incredible way, and it becomes much more difficult. It makes the picture look much more ugly. Okay, and I hope that will become more clear when I've actually drawn the picture. So, I'm going to draw a ABC transporter that is a homodimer of ABC proteins, which are half transporters. Okay, so let me draw this here. Okay, so I'm going to draw it like so. So what I'm now going to do is I'm going to go away from drawing the transmembrane domains like this as a cluster of 6 membrane spanning alpha helices, and I'm going to now represent that entire transmembrane domain as this structure here. So this is now representing the transmembrane domain. Okay. Then we have the amino terminus here coming into the transmembrane domain, and then coming out of the transmembrane domain down here, we have the nucleotide binding domain here, and then the C-terminus. Okay, so let's just color code that. So that's one half transporter protein, and now of course we know that it has to dimerize with another one that's identical to it. Okay, so let's draw the other one in here. Okay, like so. And then here is its nucleotide binding domain hanging down like so. And then here is its C-terminus, and here is its amino terminus. And then that is the full ABC transporter now, which is made out of these 2 separate proteins, which are each half ABC transporters. And I hope now you understand why the picture is far easier to draw for half transporters. If I had drawn a full transporter here, you would have expected me to then link this portion up to here, and then have the nucleotide binding domain dangling down. And it just makes the picture look so much more difficult to understand, which is why I have chosen to do it as a half transporter rather than as a full transporter. Okay. In addition, some of the main examples we're going to see are actually going to be half transporters rather than full transporters. Okay, so there are those 2 reasons. Right, so let's now talk about how this thing is going to export a substrate molecule. Okay, now we will focus on multidrug transporters now rather than normal old active transporters which just have a single substrate. Okay, and why am I doing this? Well, because we are studying multidrug transporters, but what's the difference between them? Well, the difference is in how they bind their substrate, okay, because how do these things have such broad substrate specificities? Okay, how do they have this binding site that can bind to so much? Okay, so that's one of the things that we're now going to discuss. So the mechanism is going to be very similar as to what's understood for most ABC transporters, except this binding portion is going to be specific for the multidrug transporters. Okay, right. So here we have our ABC transporter that is waiting to bind its substrate. Okay, now this site here is what I'm going to use to represent the drug binding site. So the drug is going to come and bind in between these 2 transmembrane domains. Now at the moment the transmembrane domains are in a special conformation. Okay, so they are in a conformation which is known as the inwardly facing conformation. Okay, so this is called the inwardly facing conformation. And whilst they're in this conformation, a drug molecule from the cytoplasm can come and bind here. Okay, so remember the characteristics which defined our drug molecules that could bind to these multi-drug resistance transporters. Okay, they were going to be lipophilic, which meant that they were neutral molecules. They were going to be planar, which meant that they had probably a lot of aromatic structures within them. Okay, they were going to be generally quite small, less than 800 Daltons. And they were also going to be weakly cationic. Okay, so let's remember those 4 properties as we discuss how these molecules are going to bind into this binding site here. Okay, right. Well, basically, the residues which line this binding site here are generally very hydrophobic. Okay, so the amino acid residues of the transmembrane domains which are lining this binding site that's available for the drug molecules to come and bind in, they're generally going to be very hydrophobic. Okay, so they're also going to be very neutral structures. Okay, in addition, you generally have a lot of aromatic residues as well. Okay, so how then do the drug molecules come and bind in here? Well, what is thought to underlie the binding is firstly a lot of hydrophobic interactions, okay, which remember is all about water molecules pushing hydrophobic molecules together so that water doesn't have to interact with them, basically. So hydrophobic interactions, which means that the drug molecules are going to get pushed basically into interacting with these other hydrophobic residues because the water doesn't want want to have to interact with them. Okay, so hydrophobic interactions is one of the thoughts for how the drug molecules can interact with the residues of this binding site. Okay, and then there are two other types of interactions, okay, which are a little bit more complicated to understand. Okay, so we're just going to go into this very, very basically. Okay, to understand this properly, I'm afraid you need to know molecular orbital theory, which is not something that I am particularly well up in, but all we need to know is what these interactions are and that they can exist between the drug molecule and the aromatic residues. Okay, so this hydrophobic interactions, this was all about hydrophobic parts of the drug molecule interacting with hydrophobic residues in the binding site. So I might just draw a little bit of this. So sticking out, you're going to have lots of hydrophobic residues here into this binding site, and that's going to help the drug molecule, which might be here, interact with this binding site. Okay, but now we've also got aromatic residues, which I might show in blue here, sticking into this binding site, and these are also going to help the drug molecule to bind there. Okay, now how? Well, one interaction that is believed to play a part is what's known as pi stacking. So, what is pi stacking? Well, pi stacking basically means that aromatic rings can stack on top of one another. Okay, so let me draw a picture of this. So basically, we'll go back to our favourite example of an aromatic structure, which is benzene. So let's say this is our hexagon, which is a benzene ring here. So this is representing a benzene ring. And we know, of course, that the electrons, okay, rather than being in those alternating double and single bonds, are really in delocalized rings above and below the plane of the benzene ring of carbon atoms. Okay, well, what can basically happen is you can bring in another one of these benzene rings, okay, like so. And these things can stack on top of each other and can form incredibly very strong interactions. And indeed, if you do have benzene, pure benzene as a chemical, you often find that benzene molecules end up dimerizing together and they're stacked on top of one another. Okay, so there is some powerful interaction that helps benzene rings stack on top of one another. And this interaction here, okay, is known as pi stacking. Okay, now I can't really explain to you, because I don't understand it myself, why these benzene rings stack on top of one another. Whether it's that the delocalized rings of this one below here sort of fuse with the delocalized rings of the one on top of it, I don't really know. I don't understand this. Okay, but it is an interaction that is strong, and we know that our drug molecule has aromatic structures within it, and the walls of this binding site have aromatic residues in it, so it's plausible that the aromatic rings are interacting with each other in this pi stacking way, which apparently is quite a strong interaction. Okay, so the final interaction I want to mention is what's known as cation-pi interactions. So I suppose I should just explain why this was called pi stacking. So pi refers to the electrons in these delocalized rings. So the electrons in the delocalized rings are called pi electrons. Okay, so that's why it's called pi stacking. That's where the pi is coming in here. Okay, so cation-pi interactions, this one's a little bit easier to understand. Okay, so in cation-pi interactions, the idea is that if you have a molecule which has a cationic charge, a positive charge, then this can interact with the pi electrons in a delocalized ring. So if we draw another benzene ring here, okay, so here's our benzene ring, and then you'll have the delocalized rings of electrons above and below the benzene ring here. Okay, and the idea is that these delocalized rings of electrons will have a negative charge, okay, because they contain electrons, and then of course the benzene ring in between them will have a positive charge here. Okay, and basically then what can happen is if you bring in a cation, okay, which will also have a positive charge, so let's put our cation here, that it can interact with the pi electrons in a delocalized ring, and that can form an electrostatic interaction. So cations can interact with aromatic rings through these cation pi interactions. And we said that quite a few of these drug molecules that these multi-drug transporters transport do have a weak cationic charge. Okay, so that charged portion of the molecule may be interacting with an aromatic residue in this cation pi interaction. Okay, another thing to mention is that all of these interactions are very, very broad, okay? There's no specific groups that are involved here, okay? There are loads of plausible different groups that can be involved in these interactions. So the idea is that many different molecules can interact in this binding site using these same set of binding mechanisms, basically, okay? Indeed, there is also the idea that in fact, multiple drugs can actually bind in this binding site at the same time. Okay, so what can happen is molecules, these hydrophobic molecules, you can get multiple of them binding to this binding site at the same time. So we've got one there in pink. You could also have this one in yellow binding at the same time. Okay, and then what can happen is this transporter could transport both of them at the same time, basically. Okay, so there's the idea that the binding site, because it's so unspecific, basically, it's just loads of these interactions that can form between a very broad range of molecules, that multiple molecules can bind in there at the same time because it's a large binding site, basically, okay? And then both of these molecules could be transported at the same time. Okay, so that's what I understand about how these transporters achieve this very broad specificity for these molecules that all have these basic chemical properties. It is that no particularly strong bonds are formed. Instead, loads of these bonds that are very, very broad in specificities can form between the molecule and the walls of this binding site. Okay, right. So now let's talk about how the transportation is actually going to be coupled to the hydrolysis of ATP. So what then happens is, whilst you're in this inwardly facing state, which is what we're in at the moment, okay, so I'll bring this back down a bit, we're in this inwardly facing conformation, the drug molecules will bind into our drug binding site here, okay, and then what's going to happen is two molecules of ATP are going to bind to our nucleotide binding domains. Okay, and this is going to cause the nucleotide binding domains to dimerize. So let me show this. I'd quite like to show this on the same page so that we have the two pictures to compare. Okay, so here is the cell membrane again. Okay, here is one of our transmembrane domains here. Okay, here is our second transmembrane domain, like so, and then we've got our drug molecule, or molecules, which we'll show bound here. Okay, so to make it more obvious, I'll just have one molecule bound in there at the moment, but remember that there is the very real possibility that it's just a very large binding site which can have loads of these molecules binding to it at the same time. Okay, and now what's going to have happened is the nucleotide binding domains are now going to have dimerized together when the ATP bound. Okay, so like so. Here's one nucleotide binding domain, here's the other nucleotide binding domain, and I just want to make sure this is in view of the camera. And now you're going to have two molecules of ATP which are involved in that dimerization. So there's one molecule of ATP, there's another molecule of ATP. So the molecules of ATP cause the dimerization of the nucleotide binding domains. So each one of these nucleotide binding domains ends up bound to two molecules of ATP, okay, but there are in total only two molecules of ATP. They're being shared between the two nucleotide binding domains. So don't get confused and think that there's four molecules of ATP there, there's only two. Okay, so there's one of the half transporters now complete, and here's the other one. Okay, so that then is the dimerization of the nucleotide binding domains by the binding of ATP. Now this state will only be very transient, okay, because at the moment it's still, the way I've drawn this is it's still in the inwardly facing state. Okay, in reality, when the ATP molecules bind, that causes the nucleotide binding domains to dimerize, and coupled to that conformational change is a conformational change in the transmembrane domains, and they're going to change in conformation into the outwardly facing state. Okay, so maybe I'll show this here. Okay, so now what's going to happen is the whole transporter is going to change conformation, specifically the transmembrane domains are going to change conformation so that the whole thing moves now from being in an inwardly facing state to being in an outwardly facing state, like so. Okay, so here it is in the membrane, and then you'll have the transmembrane domains dangling down, and at the moment they'll still be dimerized and they still have the molecules of ATP, so there hasn't been any hydrolysis of the ATP yet. The ATP is still alive and well and with the nucleotide binding domains. So here are the C terminals, here are the amino terminals. Okay, and when it changes in conformation like this, it's now known as the outwardly facing conformation. So it's in the outwardly facing conformation, or you can say the outwardly facing state. Okay, and when it changes conformation like this, it moves the drug molecule into the extracellular portion here, and then once it's changed conformation to the outwardly facing state like this, it then loses its affinity for the drug molecule. Okay, so the drug molecule now can no longer interact with these with this binding site, and it's believed that what actually happens that causes the drug molecule to lose affinity for the binding site is that when it changes in conformation, lots of the alpha helices, remember these transmembrane domains are fundamentally made out of alpha helices, okay, lots of the alpha helices end up spinning, okay, and what's believed to happen is they spin so that the residues that are involved in the drug binding to the binding site here are spun out of the binding site, so that now the drug molecule literally cannot interact with those residues anymore. They're now facing in the opposite way, basically. They're facing into the transmembrane domain rather than out into the binding site. So the spinning of the alpha helices that occurs in this conformational change is believed to be the reason that the drug can no longer bind there, okay, because literally the residues that it was interacting with have been spun away from it. Okay, so what will then happen is the drug will be released into the extracellular fluid. Okay, so let's now mark on the ATP molecules here in pink again, and then here are the nucleotide binding domains here in orange. Okay, like so. Right, the next thing that will happen then is that the ATP molecules will then be hydrolyzed. Okay, so remember, we've lost the drug molecule now, it's gone. We've done our job, we've moved the drug molecule out of the cell. Okay, now to return the ABC transporter back to the original state, what has to happen is the two molecules of ATP here have to be hydrolyzed to ADP and inorganic phosphate. Okay, so both of them are going to be broken down, not just one of them. Okay, if you know anything about CFTR, you'll know in CFTR, cystic fibrosis transmembrane conductance regulator, it's just one of them that gets consumed, but in most ABC transporters, it's both that get consumed. Okay, so they're both hydrolyzed down to ADP and inorganic phosphate. And then these ADP and inorganic phosphate molecules will be released, and then the two nucleotide binding domains will fall apart, so they'll no longer be dimerized, and then you'll change back in conformation to the inwardly facing state. So these molecules will have gone now, these will have fallen apart, and that will move it back into the inwardly facing state, and then you'll be back to stage one here. So this is the cycle basically, from here to here to here to here. Okay, so when the ATP is hydrolyzed to ADP and the ADP and the inorganic phosphates are released from the nucleotide binding domains, the nucleotide binding domains fall apart, and that then triggers the conformational change back to the inwardly facing states in the transmembrane domains. Okay, right. So we'll call it there for this video, and in the next video what we will do is we'll discuss examples of multi-drug resistance ABC transporters. Okay, we'll start with examples from bacteria, and then we'll look at an extremely important example in humans, which is P-glycoprotein.